Vanakkam. Welcome to this course on Classics in Neuroscience. In this series of videos, we have been looking at synaptic potentials and receptor potentials. Previously, we looked at action potential in the previous week. Since recently, we have been looking at the work of Jack Eccles. In this video, we will look at invertebrate simple systems such as Aplysia and the seminal contribution of Ladislav Tauch in identifying the Aplysia as a model system for study of electrophysiology, neurophysiology. So, Ladislav Tauch was born in Czechoslovakia in 1926 and he studied the electrical signals of plant cells, for example, until 1945, where he worked in France. But later, after that, he started studying the nervous system of the garden slug Aplysia. He identified cells in the abdominal ganglion that gave some important crucial insights into the synaptic properties and membrane characteristics. The table here presents a chronological summary of the work done by Ladislav Tauch in this field. In 1955, he established synaptic responses and basic types of induced and spontaneous electrical activity. 1956, he demonstrated IPSPs and biophysical properties, subthreshold oscillations with other colleagues. In 1957, he demonstrated the presence of electrotonic potentials. This is written as electronic, not electronic, but rather electrotonic potentials, and further demonstrated inhibitory potentials, backfiring antidromic responses. In 1958, he provided evidence for responses to injected substances, repetitive synaptic responses, interneuronal actions, and effects of hyperpolarization. In 1959, he provided evidence for existence of interneurons and long lasting hyperpolarization, a whole body of work. The first use of Aplysia as a model system was perhaps by Arvanitaki in 1941 in Marseille, which was later perhaps picked up by Ladislav Tauch, who performed a whole range of work using this model system. Why is this model system so important? Because it has a simple central nervous system, a simple brain. Let me read from one of the recounts of a colleague of Ladislav Tauch. I am just reading a excerpt. When he introduced the modern electrophysiology to study the nervous system of Aplysia, no one imagined that the simple brain of a mollusk would be so crucial to the understanding of more complex nervous systems. So, this was a remarkable pioneering effort by Ladislav Tauch. Unfortunately, the outside world was perhaps completely oblivious to this work because almost all of this work was published in French. Remember, we discussed this in our previous discussions. Is English the only language of science? We discussed this, and here is an example of a scientist disadvantaged because the work was published in a different language. This work was considered so seminal and so crucial, and Aplysia as a crucial model system was so critical that Eric Kandel, Nobel laureate Eric Kandel, who is also famously the author of the tome, the textbook of neuroscience, Principles of Neuroscience, Eric Kandel describes how he learned from Ladislav Tauch about the use of Aplysia as a model system. He describes, let me read from the text. When Eric Kandel was looking for a model system, because he was just finishing his training with Wade Marshall and K. Frank at NIH, and he was looking for a model system, he describes, He was searching for a preparation that would enable him to pursue his aim of an animal with a simple reflex that could be modified by learning and that was controlled by a small number of large nerve cells whose pathway from input to output could be identified. In that way, I could relate changes in the reflex to changes in the cells. This is what he was looking for. He essentially found what he wanted in Aplysia. So, he realized the advantages of Aplysia for doing this line of work. So, he decided to train with Ladislav Tauch to learn about how to use Aplysia for his line of work. Perhaps this led to his pioneering work on learning and memory in Aplysia. Such a primitive system showing such complex behavior or complex mechanisms in learning and memory that later went on to become Eric Kandel's life work on Aplysia. This was Ladislav Tauch's enduring impact on synaptic properties and neurotransmitter actions. Another major legacy is his inspiring Eric Kandel to work on learning and memory, because this is a very suitable model system with a simple brain, but that was still complicated enough to have advanced behaviors and show advanced mechanisms such as learning and memory. So, there was this continued search for large accessible cells for physiological analysis. Remember the squid giant axon, the neurons of Aplysia. This led J.S. Alexandrovich to study the stretch receptor cell in crayfish. What is this stretch receptor? That is the muscle spindle. Exactly how the muscle spindle works and the details my students will share with you from a previous video. Also, we will also share with you videos of how IPSPs and EPSPs work in both agonist activation, antagonist activation, and how inhibitory interneurons function. Those things my students will share the videos from a previous course on neuroscience of human movements. So, whenever a muscle is stretched, it causes an action potential in these stretch receptors. These initial studies on the stretch receptors and the potentials were conducted by Kufler, the same Kufler who worked with Eccles, I suppose, and Carlos Izaguirre, suppose he is from Chile, I think. This produced a depolarizing potential upon muscle stretch that initiated an action potential and transmitted this information to the nervous ganglion in the crayfish, and that then produced perhaps a postsynaptic potential and caused a form of information integration. Note that in this case, information is received from the receptor cells using dendrites. Previously, we have discussed how axons are communicating or crucial in communicating. In this case, information is received from the stretch receptors using dendrites at this level of organization. So, how these sensory receptors respond to stimuli? In this case, the stimulus is stretching. So, we discuss how dendrites are involved in cellular organization in next set of videos. With this, we come to the end of this video. In this video, we looked at two systems, especially the pioneering work of Ladislav Tauch in his use of Aplysia as a crucial model system that was then taken over by Eric Kandel. In one of his lectures, Ladislav Tauch essentially inspired Eric Kandel to take up Aplysia as a model system, and Eric Kandel took up this as a model system for his study of learning and memory. and J.S. Alexandrovic's work on crayfish and the stretch receptors. With this we come to the end of this video. Thank you very much for your attention.